is DNA replication. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four bases called adenine, thymine, cytosine, and guanine that form pairs between the two strands. Adenine only pairs with thymine, and cytosine only binds with guanine. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA is directional in both strands, specifically by a 5' and 3' end. This is important for replication as DNA polymerase only progresses in the 5 to 3' direction. One strand is oriented in the 3 to 5 direction, which is the leading strand, while the other is oriented in the 5 to 3' direction, which is the lagging strand. The two sides are therefore replicated in two different ways. The leading strand is the simplest to replicate. Once the DNA strand has been separated, a short piece of RNA called a primer binds to the 3' end of the strand. The primer always binds at the starting point of replication. Primers are generated by the enzyme DNA polymerase. Enzymes known as DNA polymerase are responsible for creating the new strand by a process called elongation. DNA polymerase III binds to the strand at the site of the primer and begins adding new base pairs, complementary to the strand during replication. Replication proceeds in the 5 to 3' direction on the leading strand. The newly formed strand is continuous. The lagging strand begins replication by binding with multiple primers. DNA polymerase then adds pieces of DNA, which are called Okazaki fragments, to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjoint. Once both the continuous and the discontinuous strands are formed, an enzyme called exonuclease removes the RNA primers from the original strands. These primers are replaced with the appropriate base. Another enzyme proofreads the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single united strand. In the end, replication produces two DNA strands, each with one strand from the present DNA and one new strand. This making is known as strands that are semi-conservative.
Transcription is a process by which the cells make proteins. The process begins with initiation, which is when the enzyme's RNA polymerase binds onto a part of the gene called the promoter. It is recruited by transcription factors. The RNA polymerase binds and unwinds the DNA strands and begins to read one of the bases of the DNA strand. The enzyme makes an mRNA strand using the DNA as a template. The mRNA strand is a copy of a gene and is sent outside of the nucleus. The base pairing is T to A and C to G, but on the mRNA strand there is no T, so the base pairing is U to A and C to G. The next step is elongation. During this step, it is the addition of nucleotides to the mRNA strand. The RNA molecules is produced by RNA polymerase as it reads the DNA template strand that is complementary to the mRNA strand. The polymerase will keep reading the template strand until it reaches a terminal signal, such as a poly-A signal. Termination is the end of transcription and occurs when the RNA polymerase causes a terminal signal. The mRNA strand is complete and it detaches from the template strand.
To start translation, the mRNA exits the nucleus into the cytoplasm, where the ribosome is located. Translation starts from the start codon AG, which codes for the amino acid methionine, enters the P site of the ribosome, which is where initiation begins. The ribosome takes in one tRNA at the S site, which would then bind with 3 mRNA nucleotides, also known as the codon. This new amino acid will be added to the growing strand of polypeptides next in the P site. This process will continue here. The growing polypeptide chain will remain in the P site. The tRNAs that are used exit out of the E site. The polypeptide chain over time will create a protein. After this process is complete, a stop codon will signal the ribosome to stop coding. This is called termination. The new made protein will then exit the ribosome and undergo post-translational modification.